So we're going to go into classification in action. That means actually getting out in the field and collecting some data and starting to use it to either classify organisms or ecosystems, and try and find out how we can use that classification and that analysis to understand the interactions that are happening within an ecosystem. So classification in action has an awful lot of maths, it has an awful lot of hands-on collecting data, and more than anything else, you actually have to plan your practical components. Otherwise, the data you collect, well, it's quite honestly, it will be rubbish, and you won't be able to get any decent interpretation out of that data unless you put a little bit of thought into how you're actually going to analyze a system. So let's go and have a look at a few things that we need to know about this. First of all, we have to relate back to what we've learned in math. So one of the things you did in data and statistics in math is that you learned about census and samples. So we're going to go over them again. Then we're going to look at different techniques that we have to actually collect data. So census versus sample is first. If you remember, a census is where you collect information on a whole population. And if you're collecting information on a whole population, number one, it is very time consuming, and number two, it is very costly, and it takes a lot of time to actually analyse that data. So much so that we only do a census in Australia every 5 years. It's important information that the government uses to try and figure out where resources go and where we're going in the future. However, it's a huge job to actually plan and implement and collect and then analyse that data. So census is great for detailed data, dreadful for time and money. Okay, so we tend to do a sample instead. Samples are where you collect a portion of the population, that same information from just a few individuals, and if you've selected them properly and randomly, they should give you the same average numbers as you would have got if you'd looked at every single individual in that population as well. So a sample can hopefully give us a snapshot of what the average looks like in a population if we've selected the individuals correctly. However, it doesn't if you don't plan it properly. So we're going to have a look at quality sampling and not so quality sampling in just a minute. Okay, sampling techniques is what we're going to look at then. How do we sample to get good data? The quickest method is called random sampling, where you literally just have an area, here's my area here, that we're going to investigate, and you randomly sample grids within that area. Each one of these grids would be a set amount of area, so maybe a 1 by 1 meter grid or a 10 by 10 meter grid, and you would randomly select those grids and you would count up the organisms that are in there, what different type of species they are, you'd identify that. You'd even maybe take in some other information like, are they young, are they old and mature, are they babies, what condition they're in, what abiotic factors are in there. So you can collect a huge amount of information in that one little area there to not only tell you what's living there, but talk about the factors that are in that area that might attract them to be in that little grid there. So random sampling is really quick and really simple, but however, you've got to be careful, because I've done this a million times. When you say to students, okay, go out there with your little quadrat, put it down and count everything that's in that square, they will go for the easiest place first every single time. None of them will delve into the scrub to try and find out how many grasses are in the scrubby bush. They'll always go for the easy pick. So we tend to have a lot of bias in how we choose where we're going to count. So we tend to use computers to actually lay out a grid pattern and tell us where we're going to count, so that we don't allow our own personal biases to affect that. However, as you might have seen here, there's an awful lot of stuff that's missed with random sampling. You can actually see when you look at the whole picture that there's a pattern here. Huge amount of frogs and turtles over here, so I'm guessing a water area there. A lot of birds in this band here. No frogs and turtles or birds in this area here, but none of that's really picked up by this random sampling process. So we've only got one grid that has frogs and turtles in it at all, and only a few with birds. Seems like a lot of these species are underrepresented if you only looked at just these boxes that I've shaded. So random sampling doesn't always work. It will only work if you're looking at an area that's very uniform in its distribution. So say this area here, we've pretty much got frogs and turtles in almost every one of those grids there. If we did just random counting in this little area here, it'd be fine. But when you do it across a large area like that, and it's not uniform, so we don't have frogs and turtles over here, it's not a uniform distribution. You can't use random sampling because it doesn't pick up on those sorts of patterns of where things hang out if they only hang out in certain areas of your ecosystem. So random sampling, we'd use it maybe on like a grassland or on a small area of beach where it's all the same, but we certainly wouldn't use it where we're going from a bushy area down to a creek, because animals and plants all tend to hang out in just the zones that suit their living needs. So let's have a look at the next technique, which is systematic sampling. So systematic sampling is where we have a system or a pattern to make sure that we get a spread across the whole area that we have, to hopefully catch any little patterns of distribution like I've just mentioned before. So that's systematic sampling, where you have a pattern or intervals that you actually count up in. You can do it three ways, basically, with a line, with a belt, or a grid, and we're going to have a look at each of them now. So a line is basically like this. You string a line, it doesn't even have to be parallel, some of them could go that way, and you count anything that falls on that line. So I'm going to zoom in if I can here. So essentially with a line one, we count only the stuff that falls on the line, not the whole grid, just the line. So we count that organism, they're trees by the way, that one there, that frog, that tree, nothing there because none of them, maybe he's just touching line, but that would be about that. You have a rule, basically either it's touching the line or it's not, and that's the only time you count it. So only the things that literally are crossed by the line or touch the line get counted. So line transects, the positive about them is that they're really easy to set up, really easy to get through quite quickly, but sometimes you tend to miss a lot of the details. However, very cost effective, and they will do better than random sampling. So that's the first one. Now the next one is belt. So belt is essentially, you could do this with lines, like you can string one line on this side, one line on that side, and make a belt. That's usually how they set them up, two parallel lines, and you count everything that's within that line, and also mark its position in the belt as well. Basically it takes in a lot more information, but it's an awful lot more time consuming as a line would be, as you can imagine, because even if that's a meter squared, if you're in a bush or even in grassland, there's an awful lot of species in the grassland in a 1 meter square area there. So belt is a lot more time consuming, but when you have a belt like this, you can actually start to see the trends and the patterns as you go down the belt. Now let's look at grid. Now, not all books will talk about this as a grid. They'll often say that it's a line transect, where you have a grid that you put along that line transect and you count just the grid on the line transect. So I call it differently as a grid just because it makes a bit more sense. Or you could think of it as a belt with pieces chopped out of it. Okay, either way, you don't have just a line, you don't have just a belt, you have specific intervals of quadrats. In this case it would be 1 meter by 1 meter, and they're set at intervals. So in this case it's every fourth one, and hopefully that will get a nice distribution across your entire area that you're counting in, and it will mean that you hopefully get a better picture than you would if you just had a couple of belts or a couple of lines. A grid, basically, another way of thinking of a grid is belt transects, but because you only do every second one, you can do more belts. Think of it like that. And essentially it gives you more information and a better coverage of your overall area. if you don't think about where you're placing those lines and you don't think about your intervals, so how far you're placing them apart, they're not going to give you a very good picture. So in this system here, the line, we can see that, like, there's my frogs, so my turtles there. That's almost a quarter of my entire area there. But if you looked at how many you counted up on that line, there's no way that they'd be a quarter of the species that you're actually trying to represent in that area. So you wouldn't get a very good picture. If you're going to use line transects, you'd have to use a lot more lines to get a decent picture here, because otherwise it just won't come out clearly. And belt 2. Think about the intervals. If we spread these belts right out, we could miss some important features. So notice here that this belt here completely misses any of the birds that are in this system, and if we look at the big picture, we can see there's a large range of them right there, but the actual birds you've counted up in the belts that you've placed here really doesn't give us a picture of that happening. So if they're not close enough together, or if they're not close enough together in intervals, basically, you're not going to see the picture at all. And if they're too close, then you're going to be, like if we did every second one, you're going to have so much work that you'll never get finished. So the idea is to balance it between intervals that are going to give you enough data to get a clear picture, but not take you an year to collect. So often instead of belts, we will break up these belts and do it as a grid. So this would be covering the same amount of area as my belt did, like that. However, because I interdispersed it every second one, I end up getting a much more clearer picture that allows me to get a better idea of where organisms hang in that system. Okay, so that's systematic sampling in a nutshell. But it's not always perfect. If we then take that and just do an average, so in this area here, what's my average number of turtles, frogs, birds, trees? You're going to find that the spread of how many you find in every meter squared is actually quite variable. Okay, because there are no turtles up here and frogs, but there are down here. So when you actually look at the average number of turtles and frogs per grid, and birds per grid, and trees per grid, it's going to have a huge amount of variation in the data. We call that spread. And because of that, we have to actually look at our data a little further. If we average out the results of what's in every one of these grids even, or the belts, or the lines, it's still going to have a huge spread about density of each of these organisms. That tells us that we actually have to go into this system in a little bit more detail. Really, just doing a grid pattern like this is not going to give us good enough data. We need to separate out the organisms that obviously hang out here with the organisms that obviously hang out here. My guess is that there'd have to be water here for the frogs and the turtles, but there's no water over here because there's no frogs and turtles. But there's birds hanging out a lot here, so there's either bushes or shrubs or trees here, and you've obviously got two very distinct areas with very distinct abiotic data. So one's going to have a lot more water than the other. One might have trees. The temperature would be different, the amount of water would be different, the types of bushes and shrubs in the area would be different as well, so different biotic data, because these guys hang out where they're able to survive. There's different distinct areas here, and if we just sample the whole thing and average it across the whole, we're not going to get a good picture. Because of that, we go a little bit further than systematic sampling. We then go into stratified sampling. Stratified sampling is where we identify distinct areas within the place that we're investigating, and we investigate them as a group separately. So we try and split them up. So back here, if I was going to stratify this, I would investigate this area here separately to, say, this area here, because I can see that there are at least two distinct zones, maybe even three, because there's no birds or turtles and frogs hanging out in this area. So you need to make sure that you split up those zones and then investigate them each separately using random sampling or systematic sampling.